Previously, we discussed the initiation step, and in this video, we are going to talk about the elongation and the termination step of this process. To explain the transitions, we'll talk about the promoter clearance, the formation of elongation complex, and how it transitions into termination, which depends on specific termination sequences and termination factors, especially TTF1 and PTRF. We will start where we left off in the previous video, which is the pre-initiation complex, where UBF dimer binds at the promoter and the DNA wraps around it. For simplicity, I'm only drawing out the core promoter, and at the core promoter, the SL1 factor binds near the AT-rich region. And onto this, the RNA polymerase 1 attaches, where the rRN3 interacts with both SL1 and UBF. The polymerase also carries with itself the casein kinase 2 and a topoisomerase. And this is the pre-initiation complex of RNA polymerase 1 beta. And this is what we discussed in the last video. Now, in order for the polymerase to move away from the promoter, or clear the promoter, the enzymatic factor casein kinase 2 phosphorylates rRN3, also known as TTF1A, at the serine in its C-terminal domain. This is a type of post-translational modification that we have seen in multiple other processes as well. In yeast, the CK2 also phosphorylates some subunits of the polymerase, but it doesn't appear to do that in mammalian systems as much. In addition to all this, the UBF also stimulates the polymerase 1 beta to release from the promoter. And in some cases, transcription intermediary factor 1C is also required for promoter clearance. But its exact function in the transcription is sort of unclear. In any case, all of these processes and interactions come together and release the polymerase from the promoter. And if we sketch out the resulting configuration of factors, you notice that the UBF dimer and the SL1 still remain bound at the promoter, along with the rRN3, which now is phosphorylated because of CK2. And this modified form of rRN3 is then immediately released from the promoter, and the polymerase has moved ahead where the topoisomerase and CK2 are still attached, in addition to TTF1C. Looking back at the promoter, when the rRN3 is released, the UBF and SL1 are the only factors left at the promoter, which is exactly what you need to recruit another polymerase 1 beta that can initiate a new round of transcription. So, once the polymerase has cleared the promoter, it must enter an elongation step, which also appears to depend on certain factors, like TF2F, which we saw in RNA polymerase 2 as well. But in the case of RNA polymerase 1, some subunits actually are analogous or behave, functionally speaking, like TF2F, so you don't technically need this factor. The elongation also seems to be dependent on TF2S, which is required by the RNA polymerase 2 as well, when it gets paused and needs rescuing. Specifically speaking, it is unclear what TF2S does in RNA polymerase 1 elongation. And we also don't understand the function of TTF1C. So there are a lot of things that are unclear when it comes to what factors are required and what they do in the elongation phase. Anyways, just like RNA polymerase 2, Pol1 is also dependent on the FACT complex, which is this chromatin remodeler that functions to remove histones from the chromatin. So all these factors come together and assemble onto the polymerase. And the RNA polymerase moves forward where the FACT complex removes the histone roadblocks ahead. And this is the moving elongation complex of RNA polymerase 1 beta. And by the way, the movement of polymerase itself and the synthesis of the RNA is done by the Brownian ratchet motion model that we talked about in detail when we discussed canonical transcription. I have linked that video down in the description if you are interested. And this sums up the promoter clearance step and the elongation complex formation. Now let's move on to a more interesting step, which is the termination stage. To understand termination, we must look at the ribosomal RNA gene on the DNA. We've already seen the UPE and core promoter when we discussed the initiation step, and this is followed by the ribosomal RNA genes. According to our discussion so far, the elongating polymerase starts from the start site and reaches the end of the ribosomal RNA gene. And that's where it has to terminate the transcription. Now, if you recall the concept of kinetic window that we discussed in the canonical transcription, the window in ribosomal RNA gene is about 500 bases downstream of the coding region, where you typically find a PolyT stretch. And about 50 bases from the PolyT stretch are these short identical sequences that are spread throughout. These are called Sal Boxes. Somewhere in these boxes is a region called RFB, and at the end, you often find an origin of bidirectional replication. These are something special and we'll discuss them at the end of this video. But primarily, the sequences involved in transcription termination are these PolyT and Sal Boxes. Typically, there are 10 of these Sal Boxes called Terminator 1, Terminator 2, and so on until Terminator 10. And these are spread out in the downstream region. And here's a funny little caveat. Oftentimes, there is a Sal box that is present even in the promoter. We'll touch on that towards the end as well. The length of this termination region in total is about 1,000 bases. Now, let's get into the crux of this process and start by discussing the polyT region first, which actually is a polyU sequence if you talk about the base composition in terms of the RNA. This polyT or polyU region is also called transcript release element, and the RNA version of this has the capacity to form the AU heteroduplex. We discussed this in the prokaryotic termination as well, specifically in the intrinsic transcription termination. This polyU, however, is a bit different in this case, in that there is a transcription factor, PTRF, also known as Pol1 and transcript release factor, and this binds to the polyU on the RNA. This is the same PTRF that I highlighted in the beginning. Alright, now let's talk about Sal Boxes. And we said that there are about 10 of these in humans. They can range from 10 to 30 bases in length. In humans, they are around 11 bases in length. Mice have them at 18 bases. And I think yeast Sal Boxes are slightly longer, around 25 bases. Oh, and you call these short sequences Sal Boxes because they have a Sal1 restriction enzyme site in them. Now, the special thing about Sal Boxes is that TTF1 can bind to these boxes. TTF1 stands for Transcription Termination Factor 1, specifically 1 beta. If you are reading this in yeast language, TTF1 is called REB1. Now, looking at the DNA at the end of the 28S ribosomal RNA, you have the Sal Boxes, T1, T2, and so on. And you have this polyT stretch in the DNA. And when this is transcribed, you get polyU in the RNA. And let's, for clarity, just put the RNA polymerase on top here with its polka dot elongation factors. Now, this is where things get interesting. You have this polyU sequence in the RNA, and we said that the PTRF binds at the polyU sequence. In addition, the TTF1 binds the Sal Boxes. So taken all of this together, the AU heteroduplex at the polymerase, the PTRF binding at the polyU region, and the TTF1 in front of the polymerase all block the movement of the RNA polymerase 1. And when the polymerase gets blocked, it pauses for a moment, and this makes the TF2S think that the RNA polymerase is stalled, and the TF2S or some RNase 3 cleaves the RNA and releases it. So this ribosomal RNA can then undergo its own independent processing. But the polymerase and the other factors are still attached to the DNA. And now we need to dissociate this polymerase away from the DNA. This situation is exactly the same as the one we discussed in RNA polymerase 2 transcription termination, where XRN2 is loaded at the 5' end of the RNA. XRN2 is an exoribonuclease, and it starts chewing the RNA. And SETX perhaps also joins in this process, and all of this helps to kick out the RNA polymerase from the DNA. The principle of this entire mechanism is just like the torpedo model of the transcription termination that we saw in the polyA signal and CPSF-mediated termination as well. And to learn more about the torpedo model, you can check out the transcription termination video of RNA polymerase 2. I've linked that down in the description as well. Okay, so we're not done here yet. At this point, you should have some obvious questions in your mind. One, for instance, is that we said that there are 10 of these Sal Boxes. Well, why do you need so many of them instead of having just, let's say, 1 or 2? Turns out that sometimes if Terminator 1 does not stop the polymerase can read through and keep on transcribing. So mechanisms have evolved in a way that there are now 10 terminator Sal boxes to make sure that the RNA polymerase stops. Now, we said that there is a terminator 0 at the core promoter, and these RFB and OBR regions that I said I will cover later. So let's see what is so special about them. We'll start by RFB and OBR regions. For simplicity, I'm only sketching out the regions with Sal Boxes, RFB, and OBR. The OBR stands for Origin of Bidirectional Replication. So it essentially is the starting center of DNA replication. In yeast language, it is called ARS, or Autonomous Replication Sequence. Now, starting at the OBR, the DNA polymerase can go into the upstream direction. But we know that the RNA polymerase 1 is coming down from the other end. And if these two are not stopped, the DNA polymerase and RNA polymerase will collide. Or even worse, the DNA polymerase may even stop the ribosomal RNA gene expression. To prevent this possibility, we have this RFB, which stands for Replication Fork Barrier. At the RFB, a protein called FOB1 binds, and this prevents the DNA polymerase from interfering with the RNA polymerase. This also means that it also acts like a roadblock for the RNA polymerase to move beyond this point. So neither DNA polymerase nor RNA polymerase can get past this particular point. Alright, final thing that is left on the table now is to discuss this terminator 0, which somewhat overlaps with the core promoter. To understand its function, you have to recall that ribosomal RNA genes are present in clusters. We discussed this in the initiation video as well. So a cluster essentially looks like a repeat region where you have the same ribosomal RNA gene with its promoter and terminator boxes repeated multiple times. The space between the cluster repeat is called the intergenic spacer region. Most of the time, the intergenic spacer sequence has some cryptic promoters that can recruit RNA polymerase 1. These promoters are simply called spacer promoters. So if we just zoom into a spacer promoter, it can recruit RNA polymerase 1 beta and start the process of transcription as well. But you notice that there isn't a good termination sequence present within the spacer for an efficient termination of the RNA polymerase. But instead, you get this T0 Sal box, which is within the promoter region. And this T0 box can recruit TTF1 and stop the RNA polymerase. But notice that the RNA polymerase will stop at the core promoter, which means that if this happens enough times, you will increase the concentration of RNA polymerases at the core promoter. And this is a perfect situation if you want to start the transcription from that promoter. So in this specific context, the T0 terminator box and the TTF1 factor also act like an initiation sequence and an initiation factor. This is an absolutely beautiful strategy for amplification of the transcriptional output.